Welcome back, biology, and today we're going to start talking about cellular reproduction. We've talked a lot about some just basic biology stuff, and we talked about some biogeochemical stuff. Cell reproduction is more of a biology topic. There's a lot of chemistry that gets involved, but we're not going to go so much into the chemistry. We're more going to talk about the physical process of the biology of this. So we're going to talk about how we take one cell and split it into two, and there's several different ways we do this. If you're really simple like a bacterium, you're going to use this process of binary fission. If it's one of your just everyday normal cells, you'll use this mitosis. And when you're trying to make gametes, you'll use this meiosis. And we're going to go through each of these and explain the differences between them, the similarities, and why each kind of different scenario occurs and why those different scenarios are more beneficial and the downfalls that they have. Before we get started about the new stuff, we do need to talk about some old stuff, talk about some review. Remember that when we talked about cell theory, we said that all living things come from cells, that cells are the smallest unit of life. Remember, there's single cell organisms out there that they are one cell. And then the last thing in the cell theory, and the one that we're really going to talk about today and in the upcoming days, is all cells come from pre-existing cells, meaning that you get cells that had to have come from another cell. Another thing we need to talk about is that DNA is where we store our genetic information. It's found in the nucleus or nucleoid if you're a prokaryote. Remember, it has these, kind of double helix structure here, and these different bases will store the energy in it. The information. We're going to talk about next chapter how that information actually gets transferred and what those genes actually mean. But for this one, just remember that DNA is this helical structure. So with that, we can now jump into chromosomes. And we talked about them a little bit, but chromosomes are these structures found in all cells. In eukaryotic cells, they're rod-shaped. In bacterial cells, they're circular-shaped. But during cell division, the DNA is going to be coiled very tightly into these structures called chromosomes. These rod-shaped structures made out of DNA. So we have all of this DNA here, and it's loose, and it's a lot. There's a lot of DNA in there. So what we do is we take this DNA and we'll wrap it around these proteins called histones. And as it gets wrapped around there, it gets tightly coiled, kind of like if you take a bunch of rope and you spool it up on a spool. Or if you take a garden hose and you put it on like a holder. You take this big, long, very easy to tangle piece of thing and you make it organized. That's what the histones do. But then the histones themselves will coil into these big long coils, and those coils will then fold into these structures called chromosomes. And it is important to note, and I can almost guarantee that you will see it somewhere on your quiz, is that humans have 46 chromosomes. We have 23 pair or 46 total chromosomes. All right, chromatids. So a chromosome, normally when we think of chromosomes, you think of these X-shaped structures. That is a chromosome, but each half of that is a chromatid, and each half of that is one copy of whatever genes are on that chromosome. So the reason we get chromatids and the reason that chromosomes are two chromatids is when we're getting ready for cell division, when we're taking the cell and we're splitting it into two things, we need to have two of everything before we split the cell. So we need to have two copies of the chromosome so each new cell gets a copy. The centromere is a structure to the chromosome, and it's this point where the two chromosomes are attaching to each other. So in this chromosome here, this centromere is it's close to the middle, a little bit above the middle. But the job of the centromere is to hold these chromosomes together, these chromatids together, until they separate during cell division. Now, it's a little outside the scope of this class, but this centromere can be up top, it can be in the middle, it can be down bottom, it can be directly in the middle. It can be different places, but the important thing is it's in the middle of the chromosome, and it holds the two together. There are two types of chromosomes. The first type of chromosome we're going to talk about is the sex chromosomes. And human and animal chromosomes have either sex chromosomes or autosomes. And the sex chromosome specifically determines the sex of the organism. So they have other genetic information on them, and as we talk about human genetics later, you'll find out they have a lot of diseases that they can transmit if you have one and not the other. But in humans, they're either X or Y. Females have this 2X chromosomes here, and males will have one X chromosome and one Y chromosome. And you notice the Y chromosome is a lot smaller. That'll be important when we talk about why males are more likely to have some diseases than females and vice versa. So it'll come into play later. The second type of chromosome is the autosomes. And so these two here, these are either male or female here, but the 23rd pair here are the sex chromosomes. All other 22 pair of these chromosomes we call autosomes or body chromosomes. So any cells that are produced by sexual reproduction have 2 copies of each autosome. You started out as a zygote with all you had was one cell and 46 chromosomes. You got 23 of them from mom and 23 of them from dad, one for each pair. You got one from mom, one from dad, one from mom, one from dad, so on and so forth. Now, when we talk about chromosomes, we'll say sometimes homologous chromosomes. And these are the 2 copies of each autosome. So one from mom and one from dad. They have similar size and shape, and they carry the same genes. When we take pictures of the chromosomes in the humans, we'll call it a karyotype. A karyotype, this picture here, taken on a microscope of the chromosomes inside of an individual. And this is how we first originally found the sex chromosomes. We found them in fruit flies to begin with, Drosophila melanogaster. But in humans, we have 22 pairs of autosome and 1 pair of these sex chromosomes there. When we're talking about cells, they can be either diploid or haploid. And diploid, or 2N here, means they have 2 sets of each chromosome, so they have these sister chromosomes. Most of your cells in your body are diploid. Eye cells, ear cells, mouth cells, feet cells, all of those. We'll abbreviate this one 2N. The exception in the human body are the sperm and egg cells, and we will call them haploid cells or just N, and we'll talk about those next. These haploid cells only contain 1 set of chromosomes. So in these guys here, each one of these, this egg and the sperm, only have 3 chromosomes, and they will combine to this zygote that has 6. So each one of these has half of what the new organism will be. So they have 23 total chromosomes in humans, and we abbreviate them 1N. As these two 1N cells join, they will create a 2N cell, this 2N zygote. So all cells come from pre-existing cells. We need to divide cells. It differs in prokaryotes and eukaryotes. And in each of these, though, 1 cell will become at least 2 cells. So let's start with cell division in prokaryotes. Remember that prokaryotes have a single DNA molecule. This one here. They can have these little plasmids once in a while, but for this one, we're going to look at just the DNA here. Prokaryotes are very more simple. They don't have proteins that they store their DNA around. They have 1 circular chromosome, and they go through a different process called binary fission, where this bacteria cell here will split into 2 identical offspring. And these guys are essentially clones of another. They have the same exact DNA. They have the same exact traits. There's not a lot of change from one to the other. They look the same. They're identical. They behave the same. Not as much diversity. All right, so binary fission, the first step is we have to copy the DNA. So we take this circular chromosome and we double it. So now we have 2 identical chromosomes. As this cell now has 2 chromosomes, it will start to enlarge and start to divide. The cell membrane starts to develop between these 2. It's going to pinch them apart so that they are not, we'll end up with 1 cell with 2 sets of chromosomes and 1 cell without any. So As this keeps growing, it will grow up to about twice its size, and then it will pinch itself in half to form these two identical cells. They are haploid, they only have one copy of the DNA, but these are identical. So if you and all of your prokaryote friends all have the same parent cell, you're all basically just clones of that parent. You all have the same DNA, you all look the same, you all have the same molecules. So for prokaryotes, this is a pretty simple process, which is great for them. E. coli can reproduce every 10 minutes. So one cell splits to 2, 4, 8, 16, 32 very quickly. These guys are able to grow very quickly. They're able to take over new medium and get into new areas very quickly, where humans take 16 to 18 years before we're having babies. So this is the end of 8.1. Next video will be on 8.2. But hope this is a good start, and if you have any questions, let me know.